Chapter 1. Current Advances in Gene Editing Applications in Neuroscience. CRISPR, Brain Disorders, and the Future of Neurogenetics. What gene editing is. Why the brain is challenging. Recent breakthroughs. Future directions. What is gene editing? Gene editing is the ability to precisely modify DNA inside cells. Most common tool. CRISPR-Cas9. It can remove harmful mutations, correct genetic errors, turn genes on or off. This is different from traditional drug therapy. Why neuroscience needs gene editing? Many brain disorders are genetic. Huntington's disease, spinal muscular atrophy, Rett syndrome, certain epilepsies. Traditional drugs treat symptoms, do not fix the genetic cause. Gene editing aims to correct the root problem. Challenge. The brain is difficult to treat. Barriers include blood-brain barrier, BBB, complex neural networks, risk of off-target effects, limited regeneration of neurons. Recent advances are addressing these challenges. Advance number 1. CRISPR in Huntington's disease research. Huntington's disease, caused by mutation in HTTA gene. New research. CRISPR reduces mutant gene expression, slows neurodegeneration in models. Goal: silence the toxic gene while preserving healthy function. Advance number 2. Gene editing for epilepsy. Certain severe childhood epilepsies caused by single gene mutations. CRISPR approaches modify overactive ion channels. Normalize neuronal signaling. Early animal studies show promising seizure reduction. Advance number 3. In vivo brain gene editing. Instead of editing cells outside the body, new methods. Deliver CRISPR directly into the brain. Use viral vectors, AAV. Target specific neuron populations. Less invasive and more precise than older techniques. Advance number 4. Base editing and prime editing. New CRISPR improvements. Base editing. Changes single DNA letters. Prime editing. Precise insertions or corrections. Advantages. Fewer DNA breaks, lower risk of unwanted mutations, higher precision. Important for delicate brain tissue. Advance number 5. Gene editing in neurodevelopmental disorders. Research in autism spectrum disorders, Rett syndrome, Fragile X syndrome. Goal: correct early gene expression abnormalities. Still early stage but promising. Advance number 6. Parkinson's disease research. Gene editing approaches aim to modify dopamine-related pathways, protect neurons from degeneration, study disease mechanisms, helps identify new therapeutic targets. Advance number 7. CRISPR for brain cancer. Glioblastoma. Applications include editing immune cells to target tumors, disrupting tumor growth genes, studying tumor resistance mechanisms, still experimental but rapidly progressing. Advance number 8. Precision delivery systems. Major innovation. Engineered viral vectors. Nanoparticles crossing the BBB, cell type specific targeting, Delivery remains the biggest challenge, but progress is accelerating. Ethical and safety considerations. Key concerns. Off-target effects. Long-term safety. Germline editing risks. Brain function alteration. Strict regulation is essential. Neuroscience editing must prioritize safety. Future outlook. We are moving toward precision neurogenetic medicine, personalized brain therapies, earlier intervention in genetic disorders, combined gene editing plus AI diagnostics. Gene editing may transform treatment of neurological disease in the coming decades. In the next chapter, we are going to explore 10 recent neuroscience breakthroughs. Chapter 2. Top 10 recent neuroscience breakthroughs. How brain science is changing medicine. How AI is transforming neuroscience. What new discoveries mean for the future. Why neuroscience matters. The brain controls thoughts, controls emotions, controls memory, controls movement, defines personality. Understanding the brain helps us treat Alzheimer's, treat depression, treat Parkinson's, understand learning, build brain-inspired A. We are living in a golden age of brain research. Breakthrough number 1. High-resolution brain mapping. Scientists can now map individual neurons, neural connections, connectomy, brain circuits in unprecedented detail. Why it matters. Helps understand autism. Helps understand schizophrenia. Reveals how networks process information. The human brain has approximately 86 billion neurons. Mapping them is revolutionary. Breakthrough number 2. AI decoding brain signals. Artificial intelligence can now interpret brain activity patterns, translate brain signals into text, reconstruct images seen by participants. Applications. Communication for paralyzed patients. Understanding language processing. Brain-computer interfaces. This is not mind reading, but signal decoding. Breakthrough number 3. Advanced brain-computer interfaces, BCTIs. Implanted or wearable devices can restore movement in paralyzed patients, allow control of robotic limbs, enable communication without speech. Example: signals from motor cortex to computer to robotic movement. Future impact. Stroke rehabilitation. Spinal cord injury treatment. Breakthrough number 4. New Alzheimer's therapies. Recent developments. Targeted antibodies against amyloid proteins. Slowing cognitive decline in early stages. Better biomarkers for early detection. Early detection is key. Brain changes start years before symptoms. Research now focuses on prevention, early intervention. Breakthrough number 5. Psychedelic research in mental health. Controlled clinical trials show that psilocybin reduces severe depression, and the effect may last months. Why it matters. These treatments may help patients resistant to traditional therapies. Research is highly regulated and medically supervised. Breakthrough number 6. Brain plasticity in adults. Old belief: the adult brain cannot change much. New evidence: the brain remains adaptable throughout life. Findings show new synaptic connections form. Learning reshapes neural circuits. Rehabilitation can rewire the brain. This changes how we approach aging and recovery. Breakthrough number 7. Gut-brain connection. Advances. Scientists discovered gut bacteria influence mood. Microbiome affects anxiety and depression. Inflammation links gut and brain. Implications: new treatments for mental health. Diet-based interventions. The brain does not work alone. It communicates with the body. Breakthrough number 8. Real-time brain imaging advances. New imaging techniques allow faster functional MRI, better visualization of brain activity, improved detection of neurological disease benefits, personalized medicine, earlier diagnosis, more precise treatments. Breakthrough number 9. Neural basis of consciousness. Recent studies identified brain networks linked to awareness, thalamus-cortex communication patterns, electrical signatures of consciousness. Applications: understanding coma, improving anesthesia, studying disorders of awareness. The question, what is consciousness, is now scientifically studied. Breakthrough number 10. Gene editing and brain disorders. CRISPR-based research is being used to study genetic causes of epilepsy, model neurodevelopmental disorders, explore gene therapy for rare diseases. Future direction: precision neurology, treating brain disorders at the molecular level. What these breakthroughs mean for the future: we are moving toward personalized brain medicine, AI-assisted diagnosis, neural enhancement technologies, early detection of disease, brain-machine integration. Neuroscience is merging with AI, genetics, and biotechnology. Final takeaway: the brain is no longer a mystery in the dark. We can map it, decode it, interface with it, treat it more precisely. The future of neuroscience will transform medicine, technology, and human potential. Subscribe to stay updated on the latest discoveries shaping our future. Thank you.